That's a very easy understanding what is reduction, what is oxidation. So the question here will be, what is reduced product? What is oxidized product? And very simple, when C is A gains electrons, so C is what? Reduced product, that is C. Therefore, D is oxidized product. And in the chemistry, you also learn what is reducing agent. What is that? What is reducing agent? It's a B, is that right? Okay, what is the oxidizing reagent? It's A. Is that correct? That's very simple. We talk about in the chemistry. And that's oxidation reduction. Now, Scott gave you these slides and also these slides and talk about this here. I would directly jump into another topic, is talk about the electron transport chain and everything we could talk about together. So we're going to jump into a topic called the electron transport chain. So electron transport chain. That is ETC. Okay, let's just draw ETC, what it looks like. So all the chemical ingredients you should know. So we go NAD, go to FAD, go to FMN, and FES, usually a complex, go to coenzyme Q, go to cytochrome cytochrome C, and then you go to cytochrome A, then go to cytochrome A3, and finally is oxygen. And then the electron will be transferred, so we have this going to be transferred. So we do this all the time. This is a very standard electron transport chain. Now, obviously, different bacteria is different, especially like soil bacteria is different. Okay, that's the electron transport chain. We want to talk about several things. We're going to draw here. We're going to talk about the other side. So we write the chemical. And first of all, the first question. Where is the electron transport chain located? For bacteria, it's a prokaryotes. Cytoplasma membrane. Okay, how about fungi? Easternmost. It's eukaryotes. It's the same as you learned in biochemistry class. It comes from mitochondrion. That's a fungi. Okay. The second question. What are these chemicals? Why the order is like this? Okay, you know that. Nicotidine dinucleotides. Adenine dinucleotides. Adenine dinucleotides. What is this guy? Valvine adenine dinucleotides. What is this guy? The ferric sulfide coenzyme Q cytochrome C. Oh, you know these things. Why the order like that? This is coming to a very easy concept, which is the electron transfer. They always from is electron donor to electron acceptor. even though it is a good electron donor, it is a good electron acceptor, this rely on a 2-half reaction, which is carried by value of E. And the value of E, originally, it is related to the delta G. That equals delta, the formula should be that equals NF delta E. That's called the Farada constant. So we just keep talking about this, okay? That's delta E. So they're using two-half reaction to get the voltage value. And what they found, the more negative, the more potentially an electron donor. And the more positive, and the more potentially an electron acceptor. So they got those voltage value. How they get it? NAD plus with this guy, with this, NADH2. This voltage value is very low. Is minus 0.32. And how about this oxygen here? This is oxygen with this becomes water. This is positive 0.82. That's the reason why the order is like this. Now, all these things, you have NAD, you have FAD, cytochrome C, all these things. They are transferring electron. At the same time, they can be a donor, they can be an acceptor. So those things, we all call it an electron carrier. And there is a very important thing we want to mention, is the electron carrier must be recycled during metabolic process. When we talk about the fermentation on Thursday, we will explain this. If the electron carrier is stopped there, which means NAD become NADH2, the NADH2 couldn't go back. And this is an empty vessel, looks like here, could come in electron. If this is not recycled or stopped there, that means bacteria will be dead. So it's a very interesting topic when we talk about that. Okay, so that's something I want to mention. Now, we want to talk about something else is, we mentioned about substrate level phosphorylation, which based on the higher phosphate potential transferring power chemical. Let's say phosphoenolpyruvates. Let's say 1,6,1,3 biphosphogycerate. We just mentioned about that. But more often for bacteria, in the aerobic environments, what they do is they're using another different way to generate energy. That is called proton motive force. So what this means, motive force. That's PMF. So this is the electron transport chain. from here. Okay, NAD, FAD, FES, covalent and Q go to the oxygen. So they transfer it. Okay, at the same time, what else will happen? Let's say that this is a cell membrane system. At the same time, coupled with electron transfer, the proton will be repelled outside cell membrane system. And they will be accumulated. When all is accumulated, they create a potential energy. And once they accumulated more and more, they always have a potential they're going to go back. Now, luckily, they can do that. Because remember, in the cell membrane system, you have those integral protein. When we talk about the fluid mosaic model, you have those protein like a bridge. So these guys can go back. And when they go back, what they triggered? They triggered ADP with phosphates becomes ATP. Now in biochemistry class, you finished talk about here, but in microbiology class, we're going to have to one more thing. This, if they do the research in Proteus vulgarius, they find there is one thing is spin, is flagella. The flagella is spin. That is a true motility. And this whole thing is called proton motive force. This is Mr. Peter Mitchell. In 1920s, he found it. And in 1950s, he won the Nobel Prize because of this theory. And this whole theory, in other words, talk about oxidative phosphorylation. That's what it is. So, if you say substrate level phosphorylation is without electron transport chain create energy, that is just an excuse. It's not related at all. Because this is what we talk about is electron transfer related to the proton accumulated and then generate a potential energy, then they will come back because they always have to keep a balance and activate the ATPase. And then the ADP with phosphate becomes ATP and then trigger the flagella moving. And the spin of the flagella becomes the true motility of a bacterial cell. And this is called proton motive force theory. And in other words, chemosmosis. Is that right? That's what it is. This is the whole thing explaining oxidative phosphorylation. Okay? So, we had a couple of slides here. You could be explained to you. This is an oxidation reduction reaction, redox reaction. This is, we already talked about oxidation reduction. Lost electron is oxidation, gain electron is reduction, and we have an example for that. This is a 2-half reaction voltage value, and you can see NAD, nicotinic adenine dinucleotides, is minus 0.32, water 0.82. And you can see, basically the order is coming from more negative, go to positive. The more negative is potential electron donor, and the more positive is an electron acceptor. And we have the conclusion sentences right here to you. Okay? And this is another one, which gives you more detail on this voltage value. And this voltage value I just mentioned, it could be due to calculation with a Faraday constant. This is about, this value is a fixed. This guy is fixed at about 23.06-ish kJ per mole. They give you delta G. Okay, so it's still related to go back to the Gibbs free energy equation. It's just not many people mention about that one. Then we talk about the electron transport chain. And their location, the mitochondria, and in the eukaryotes and the plasma membrane for bacteria and archaea cells. And we give the examples of the electron carrier. It is a very good conclusion. That table is important to talk about electron carriers, the positive, their NAD, FAD, NADP. NADP we didn't mention too much because this is basically using the Calvin cycle. And like I said, I'm not going to mention anabolism because our time is limited. But this table tells you important, these are the examples of the electron carriers. And remember, we mentioned very important, the electron carrier has to recycle. Okay, if they are not recycled, then the bacteria will be died. And this is created by Peter Mitchell in 1930s. Talk about chemosmosis, which is an explanation of the ATP generated related to the electron potential proton motive force, and related to ATP synthesis, and also related to the flagellar rotation, the spin of the flagella. Okay, that's an example of the potential energy become a kinetic energy. So that's why this is very important. Next one, and the last one of this section, we want to very briefly mention the enzyme, because you already know. We want to just be very briefly, using 10 minutes to 15 minutes, we're going to talk about the enzyme. We will talk about the metabolic cycle on Thursday, fermentation, glycolysis, and also the citric acid cycle. And more important is for the ATP calculation. Now we can do some of the conclusion here. The metabolic pathway, it could be linear, and you will know this is fermentation. Usually it's linear. It could be a branch, and the branch we mentioned about the glycolysis, like that. This is a branch. It could be a cycle or cyclic. So TCA cycle, like. Okay, this is like glycolysis. So that's all the metabolic pathway. Now bacteria in a very short life, they need something to let the biochemistry reaction happen more efficiently. Therefore, it comes out with the one chemical called enzyme. And the enzyme, basically, you have a substrate, and we will generate a product. And this will let it happen, so we put an enzyme there. Now, enzyme basically is a protein. We know that. The major part is a protein. Now, what is really the enzyme for? So, for example, we want the free energies right here, and this is a reaction progress. And originally, we needed this type of energy that happen. If we use enzyme, the energy will go down. So this will be lowering down energy required for enzyme, required for substrate. Okay, so that's kind of with the function of the enzyme. Now, how do we know it is an enzyme? And I always say, if you have an ASE, you know it is an enzyme. So for example, urease, oxidase, catalase, deaminase. When you see ASE, it's an enzyme. Now, what factors will be impact the enzyme? That's very simple. Everybody knows. The major for bacteria, pH, temperature, substrate concentration, and also environmental factors. Is that right? All kinds of things, heat sensitive, and that's all kinds of things could be affected. Majorly for bacteria, pH, temperature, substrate, and other environmental factors. Now, we also want to mention about, I said enzyme basically is a protein, the major part, but does not mean it's only the protein. So most of the enzyme, what we talk about in the biochemistry reaction, this is we usually call, it is a hollow enzyme. There are two parts. The apple enzyme is a major protein part. Okay, and there's another part we call it is non-protein. We call it a cofactor. So cofactor is non-protein side. Now the cofactor can also separate into two. If it's loosely attached, we call it coenzyme. If it's closely attached, we call it prosthetic group. So these are the things we just do the review. And then it comes out another very tricky question. Substrate concentration, x-axis, y-axis velocity, and you have a curve, which is very interesting curve. Look at this, like that, and what we are looking for in this Km value. So Km equals what? Vmax. So V equals Vmax S Km value add S. So what this Km means, my call is constant. This is a concentration of the substrate required to reach half of the maximum speed of enzyme reaction. Is that right? So that's something in the chemistry we talk about. And then we also talk about, this is a lock key model. So enzyme and the substrate, they like a key and a lock, they work together. You open it, then they start to have reaction. And this is what we talk about in bacteria. We basically talk about temperature. That is why the optimal temperature for bacterial growth is always 37, because their enzyme activity is maximum. And when we make the media, we always want the neutral pH, because optimal at 7.0. But of course, it's related to their substrate products, enzyme concentration will also affect that. And then we talk about the competitive inhibition. And we talk about the competitive inhibition. So this one we mentioned about in the biochemical reaction. So what happened? You have PABA. You have something like that. Okay, what is this called? Benzoic acids. COH, sorry. Okay, you have a benzoic acids like that. PABA this is. And you have a sulfur drug. What's a sulfur drug looks like? You have a sulfur drug. And the morphology, the structure looks very similar to the PABA. So instead of the substrate reacting with PABA, the sulfur drug will attack the area or grab the area. So what happened? The folic acids will not generate. And the sulfur drug, it's a very stable one. You look at here, the bonds. The carbonyl bonds is double bonds. It's very stable. But sulfur drug, we also mentioned that this is only for inhibit the bacterial growth. Okay, it's a competitive inhibition is still reversible. But sometimes the inhibition is not reversible. If we're using mercury, like heavy metals, the reaction will not be reversible. But most of the time, what happened is allosteric reaction. So you look at the table there, or look at the figure there, there is an enzyme. Something attacking at the back of the enzyme. They change the morphology. So let the substrate do not attach to the area. Then stop the reaction. And what is the phenomenon of this guy? The allosteric regulation, it will be talk about the lacto-operon. The lacto-operon is a good example of the allosteric regulation. Okay? Last one, feedback inhibition. So you always remember the bacteria has to keep the balance. And there's a substrate, there is an enzyme, there's end products. If the end products is too much, they will transfer the signal, could be in the middle, could be in the intermediate products, it also could be in the top. They will say, please stop supplying anymore because it's enough. Because bacteria have a very short life, they have to keep everything in an economical way from the energy standpoint to survive in various environments. Okay? So this is what we have for our opening remarks of the biochemistry.